Chapter 10, The Structure and Function of DNA, Part 2. What is a mutation? A mutation can be defined as any change in the sequence of the nucleotide of a cell's DNA. Okay? And mutations can involve either big regions in the chromosome, that means a long segment of the DNA, or just one single nucleotide pair. Example is sickle cell anemia disease. So in general, mutations within a gene can be divided into two broad categories. Number first is nucleotide substitutions. That means one nucleotide gets replaced by another. And the second one is a group called nucleotide insertions or deletions. It's just like you add something or you delete something. This slide shows the molecular basis of how a single nucleotide mutation can cause sickle cell anemia. On the left side, you see a normal hemoglobin DNA in blue. It goes, the sequence is CTT. If there's and then the mRNA that will be formed is GAA. You see the complementary base pairing, and so the amino acid that comes, corresponding amino acid, is a glutamic acid or glutamate, and this is normal hemoglobin. But on the right side, you see what happens when a mutation occurs. So instead of the CTT, because of a mutation, the T in the middle has been substituted by an A. So it becomes CAT. Now, the complementary base pairing, according to that, the mRNA that is made will be GUA. And since it is not GAA anymore, this changed mRNA leads to a different kind of hemoglobin. And now the hemoglobin that is formed is valine. It's not glutamic acid. And because now the hemoglobin gets a different amino acid, the protein structure is different. So this particular, the RBC becomes sickle cell shaped. It's sickle shaped, and this RBC is not a biconcave structure, and this gives rise to a genetic disorder called the sickle cell anemia. Now let's see the different types of mutations. The first one is a missense mutation. It is a point mutation, that means a single nucleotide change, where a single nucleotide change causes a change in the amino acid. So one amino acid gets replaced by another. Then that one is a missense mutation. The second type is a nonsense mutation, where actually because of a mutation, the amino acid changes not from one to another, but it changes an amino acid codon into a stop codon. So the protein gets stopped there. That is a nonsense mutation. The third type is where mutations are involving deletion or insertion of one or more nucleotides in a gene, and where this one or more is not in a multiple of 3. So then this leads to a frame shift mutation, and these have disastrous effects because these will change the entire nature of the protein after the mutation, because you see codons occur in groups of 3. So when you have changed that, you just added 1 or 2 or deleted 1 or 2, you change the entire reading frame. So now the entire protein gets disrupted. This figure shows 3 different types of mutations and their effects. The DNA nucleotides are shown in blue, the RNA structure in red, and then the amino acids in purple. So if you look at number A, that is base substitution, where because of a change in the mRNA, now the amino acid has changed from glycine to serine. The second one, B, is a nucleotide deletion, where one of the nucleotides got deleted, so you see the change in the amino acid sequence. While the third one, C, is a nucleotide insertion, where when new nucleotide was added, again you look at the change in the amino acid sequence. How do mutations occur? They can occur in a number of ways. They could either occur naturally, so that's a spontaneous mutation. It could occur because of random errors during replication or recombination. So whenever DNA is replicating or transcription, something goes wrong naturally, and that's a spontaneous mutation. And the second method by which mutation can occur is when it is induced by a physical or a chemical agent, and then those agents are known as mutagens. So the most common physical mutagen is X-rays, high energy radiation, UV light. Chemical mutagens are so many different types, right? And some of these mutagens can also act as carcinogens. That means they can cause cancer. So we should try to minimize our exposure to such mutagens. So the question is, does mutation always seem to be harmful? No. Mutations are very natural events, and actually without mutation there will be no evolution. Mutation is the raw material of evolution. So mutations can be beneficial, they can be harmful, or they can be neutral. There can be no effect. So mutations are a rich source of diversity. Okay, if there was no mutation, there would be no diversity of genes, and then the natural selection will not act. Now let's look at virus and some other non-cellular infectious agent. What is a virus? In very simple words, a virus could be considered as a particle having genes in a box. Why? Because there's a nucleic acid, either a DNA or an RNA, and it is wrapped around by a protein coat. And in some cases, there might be an envelope too. So viruses are in the boundary line between living and non-living. They cannot be considered totally living because they do not have cells and they cannot reproduce on their own, but they do resemble living organisms in that they have genetic material in the form of nucleic acid. So as I said, the virus could have a DNA or an RNA. It could be single-stranded or it could be double-stranded. The figure here shows an adenovirus with the DNA in the core and then the protein coat outside. Just like viruses can attack human beings and other animals, similarly, viruses can also attack bacteria. And then those viruses are known as bacteria eaters, or scientifically they are called bacteriophages, or simply phages for short. So the bacteriophage consists of a molecule of DNA and it might be covered by a protein coat. Once they infect a bacterial cell, most bacteriophages, they would enter a reproductive cycle called the lytic cycle. That means they make baby viruses and they just totally lyse, they burst open the bacterial cell and the viruses, the baby viruses just, they move out. But then some viruses will not kill the bacterial cell immediately. They can reproduce, but they will not lyse the bacterial cell. And then that cycle, or that is an alternative route, that is known as the lysogenic cycle. This figure shows how a bacteriophage infects a bacterial cell. In this figure, the brown colored part is the bacterial cell, while what you see above those little robot-like things are the bacteriophages, about 200 nanometers tall. They have a head and the tail, and you see a bacteriophage inserting its DNA in the bacterium. This figure illustrates the two alternative reproductive cycles in bacteriophages. On the left side, you see the lytic or the lytic cycle, where the phage will reproduce and lyse or break open the bacterial cell, while on the right side is the lysogenic cycle, where the phage incorporates itself into the bacterial DNA and will keep propagating its DNA along with the bacterial DNA each time the bacteria reproduces. And then suddenly, it would just decide to go into a lytic cycle and it would break open or destroy the bacterial cell.
In contrast to the lytic cycle, the lysogenic cycle reproduces the viral genetic material without destroying the host. The lysogenic cycle of phage lambda begins when a phage binds to the surface of a host bacterium. The phage injects its DNA into the host cell, leaving the empty protein coat outside. The viral DNA is incorporated into the host cell DNA, where it is called a prophage. Every time the host bacterium reproduces, it replicates the phage DNA along with its own and passes the copies on to daughter cells. Occasionally, the phage DNA exits the bacterial chromosome and initiates a lytic cycle. The viral DNA takes over the metabolic machinery of the host cell to make phage DNA and proteins. The host cell lyses, releasing phages which can infect other cells. This diagram summarizes the lysogenic and lytic cycles of phage lambda.
There are several viruses that affect plant, and then they cause a stunting of the plant growth and also decrease the crop yields. Most of these viruses are retroviruses. That means they have RNA as their genetic material and not DNA. Many of them, like the TMV or the tobacco mosaic virus, these are rod-shaped and there's a spiral arrangement of proteins which is around the nucleic acid. These diseases have no cure, so instead scientists and agriculture specialists, they focus on prevention because prevention is better than cure. So preventing infection and genetically modifying crop plants that are resistant to the viral infection is the only solution for now. This figure illustrates the spiral arrangement of the proteins around the RNA in the tobacco mosaic virus. In animals, there are several diseases caused by viruses, and the viruses that infect animals also, like most other viruses, they have a nucleic acid or material, DNA or RNA inside, surrounded by a coat, and that outer envelope is made up of phospholipid, which has projecting spikes made up of protein. And this particular outside envelope, it helps the virus to enter, that means infect and leave a host cell. Many viruses, for example, those that cause flu, common cold, measles, mumps, AIDS, polio, they are retroviruses. Diseases caused by DNA viruses are hepatitis, chickenpox, and herpes infections. Animal viruses have become very important in the present context because of the COVID-19 pandemic that is going on. This is what an influenza virus looks like. You look at the RNA in the center with the protein coat surrounding it, and then the membranous envelope with the protein spikes. This figure illustrates the reproductive cycle of a typical RNA virus that is enveloped. This is the mumps virus.
A virus is an intracellular parasite that can reproduce only by taking over a host cell. A virus consists of a nucleic acid genome enclosed in a protein shell called a capsid. In the virus shown here, the genome consists of DNA, but some viruses have RNA. Some viruses are also covered by a membranous envelope that is derived from the membrane of the host cell. There is usually a lock and key fit between the proteins of the capsid and receptors on a particular type of host cell. The virus attaches to a host cell and viral DNA enters the cell. The viral DNA uses nucleotides and enzymes of the host cell to replicate itself. The viral DNA then commandeers other host cell materials and machinery to transcribe its genes into messenger RNA and translate the RNA message into capsid proteins. Viral DNA and capsid proteins then assemble into new viruses. Mature viruses leave the host cell, often destroying the cell in the process. The viruses can go on to infect other cells, spreading the viral infection.
Some animal viruses reproduce in the cytoplasm, while some reproduce in the nucleus. Herpes virus, which can cause chickenpox or shingles, cold sores, and genital herpes, these are DNA viruses that actually go inside the host cell nucleus, and they get their outer envelopes from the cell's nuclear membrane. Copies of this particular virus, like herpes virus, they remain behind in the nuclei of certain nerve cells where they can remain dormant or latent for relatively long periods, and every now and then they become active and they produce the symptoms of the disease. How much damage a virus can cause depends on the patient's immune system, how quickly the immune system responded, and how efficient the repair mechanism in the tissue is. We usually completely recover from cold and cough because our respiratory tract is very, very efficient when it comes to repairing process. But in contrast, the polio virus, which attacks neurons, our nerve cells, the nerve cells do not divide. After we are 2 years of age, 24 months, after that, there is no regeneration, no mitosis in neurons. So these are not replaceable. So the damage to the neurons by the polio virus is permanent. So the only thing we can do is to prevent the disease with vaccines. Let's look at the case of the West Nile virus, which first appeared in US in 1999. Most people who get this virus, they are asymptomatic, they do not become ill. But in some cases, the virus can actually cause fatal swelling of your central nervous system, that means the brain and the spinal cord. And mosquitoes are the vectors for this disease, that means they spread the disease. There is no cure, no therapeutics for this particular disease. Now, scientists made an RNA vaccine which used a gene from the West Nile virus. Then they wanted to test the efficacy of this vaccine. They gave the vaccine to cats in 3 different doses, first in a very high dose, low dose, or placebos, that means those who were not given the vaccine. Dogs were also used and they received only low doses or placebos. After getting the first dose of the vaccine, all these animals, they received a second booster dose 28 days after their first dose. And then all of these animals in these 5 different groups, they were exposed to mosquitoes which were carrying the West Nile virus. And then they were tested for the presence or absence of infection. This slide shows how the testing of the RNA vaccine was carried out against West Nile virus. And the results showed that you can see in the table, in the placebo group, a high percentage of both cats and dogs, they got infected by the West Nile virus. In the low vaccine dose, none of the dogs got infected, but some cats did show the infection. For the high vaccine dose, none of the cats got infected. The dogs were not tried on the high vaccine dose. All of you must have heard of AIDS. AIDS is a devastating disease. The full form is acquired immunodeficiency syndrome. It is caused by a virus. The name of the virus is HIV, human immunodeficiency virus. And you know that a person, when he or she is HIV positive, doesn't mean that the person already has the disease. Just being an HIV positive does not mean having the disease. It's only when the virus becomes very active and starts producing the symptoms, that is when the person is said to have the disease. HIV is a retrovirus. It is an RNA virus. It is called retro because it reverses the flow of information. How we know the flow of information goes is from DNA to RNA to protein. But because these viruses, their genetic material is RNA, they use a particular enzyme called reverse transcriptase, which catalyzes reverse transcription. So that means they synthesize DNA from RNA using this particular enzyme. This is how the HIV or the AIDS virus looks like. This slide shows the behavior of the HIV nucleic acid in an infected cell. On the right side is a white blood cell that has been infected by the red dots, which are HIV viruses. We are going to learn more about this in the next animation slide.
Understanding the structure and life cycle of HIV, the AIDS virus, is critically important to dealing with the disease. HIV is covered by an envelope derived from a host cell membrane. Glycoproteins studding the envelope recognize and bind to receptor molecules on the host cell. A protein coat surrounds the viral genetic material, which consists of 2 molecules of single-stranded RNA. The enzyme reverse transcriptase enables the virus to make DNA from an RNA template, a trick for which this group of viruses is named retroviruses. The RNA molecules of HIV enter a host cell when the virus fuses with the plasma membrane and the coat proteins are removed by enzymes. Reverse transcriptase catalyzes the synthesis of a DNA strand complementary to the viral RNA strand, and then a second DNA strand complementary to the first. The double-stranded DNA is incorporated as a provirus into the host cell's chromosomal DNA, where it may lie dormant for years. Occasionally, the provirus is transcribed into RNA. This RNA serves as both messenger RNA for the formation of HIV proteins and as genetic material for the next generation of viruses. Protein coats form around viral RNA and reverse transcriptase molecules. Viruses bud from the host cell, acquiring envelopes as they leave.
The problem with the AIDS virus or the HIV virus is that it attacks your immune system. So it attacks your soldiers, the white blood cells, and makes them weaker and also makes them inefficient, so that the body is rendered defenseless against any other infection. And most people who get infected by this virus, they eventually die of pneumonia or some simple viruses that can cause cold and cough. As of yet, there is no cure for AIDS. We only have medicines that can slow down the progression, and there are 2 categories, 2 groups of therapeutics that are being used. The first one is the first type of medication. It inhibits the action of enzymes called proteases, which help produce the final versions of the HIV protein. The second category of medicine, which includes the most commonly known AZT, this stops the action of HIV enzyme reverse transcriptase. This is AZT drug and the T nucleotide. What works best against AIDS or for HIV infected people is a drug cocktail. And this mixture of drugs contains both the reverse transcriptase inhibitors and protease inhibitors. And this is, data has shown that HIV infection can be lowered by 80% with the proper treatment of it, the cocktail of drugs. Prions are misfolded proteins that behave as infectious disease-causing agents. They usually cause a lot of brain diseases in animal species, not only just humans, other animals as well. So while a virus DNA contains DNA or RNA, this prion consists exclusively of a misfolded form of a normal brain protein. So individuals can have in the cell, they could have normal version of the protein, and then one protein becomes misfolded, and then that misfolded protein starts behaving as if it's infectious. So any normal protein that it touches, it keeps changing it into a prion. It's a keeps converting them into misfolded proteins as well. And the only hope for developing effective treatments against these prions is to understand the process of infection. So mechanistic studies are still going on. Viruses have become very important these days. Those viruses that suddenly come to the attention of medical experts, those are known as emerging viruses. Examples could be the COVID-19 virus, the Zika virus, the West Nile virus. All of these viruses, they persist, at low levels for several years, and then every now and then they have an outbreak. How is it that they suddenly, have these outbreaks? It's because these viruses, they do not stay the same. They keep mutating. Sometimes we do have zoonotic transfer, like in the case of COVID-19, where the virus was not actually a human virus, it was in another animal species, and then it somehow accidentally came into the human and mutated to make human beings its possible host. New viral diseases arise from spread of existing virus species. This is the zoonotic transfer that I was talking about. Or viral diseases from a small isolated population can also spread to bigger populations because travel has become so easy these days, and these can cause epidemic or even pandemics. This slide shows you a sample of the major emerging virus outbreaks in the past 100 years. You can see all the important names there: AIDS, H1N1, Zika, Ebola, SARS, West Nile virus, and we can also add COVID-19, which stands for coronavirus disease of 2019 caused by the SARS-CoV-2 virus to this list. Whatever we've learned in this presentation is very beautifully summarized by the Nobel Prize winner, Joshua Lederberg, who said, "We live in evolutionary competition with microbes. There is no guarantee that we will be the survivors." And that's true. For years after years, there's a constant battle going on for survival. This is between the human body and all those different invaders, possible agents there. It could be against viruses, bacteria, it could be against protozoans, other fungal spores, and so on. So every time we recuperate or we recover from an infection, it was because the battle was won by our immune system.